Chapter 19 Sex and Genetics. Individual cells reproduce by duplicating their DNA and dividing in two. This basic process occurs in all living species, in the cells of multicellular organisms as well as in free-living cells such as bacteria and yeasts, and it allows each cell to pass along its genetic information to future generations. Yet reproduction in a multicellular organism, in a fish or a fly, a person or a plant, is a much more complicated affair. Multicellular organisms go through elaborate developmental cycles, in which all of the cells and tissues and organs of the individual must be generated afresh from a single cell. And this initial cell is no ordinary cell. It has a very peculiar origin. For most animal and plant species, it is produced by the fusion of two cells, derived from two separate individuals, a mother and a father. As a result of this fusion, a central event in sexual reproduction, two genomes merge to form the genome of the new individual. The mechanisms that govern genetic inheritance in sexually reproducing organisms are therefore different and more complex than those that operate in organisms that pass on their genetic information simply via cell division. In this chapter, we explore the cell biology of sexual reproduction. We begin by discussing why organisms bother with sex, and we describe how they do it. Generation of the special cells that carry genetic information from each parent involves a specialized process of cell division called meiosis, and we review the mechanics of that process. We then discuss how Gregor Mendel, an Austrian monk preoccupied with peas, deduced the basic logic of these genetic mechanisms. Finally, we describe how scientists can exploit the genetics of sexual reproduction to gain insights into human biology, human origins, and the molecular underpinnings of human disease. The benefits of sex. Most of the creatures we see around us reproduce sexually. However, many organisms, especially those invisible to the naked eye, can reproduce without sex. Bacteria and other single-celled organisms most often reproduce by simple cell division. Figure 19.1. Many plants also reproduce asexually, forming multicellular offshoots that later detach from the parent to make new independent plants. Even in the animal kingdom, there are species that can reproduce by budding. Figure 19.2. And some worms can split into two halves, each which regenerates its missing half. And the females of some species of insects, lizards, and even birds can produce eggs that develop parthenogenetically, that is, without any need for males, sperm, or fertilization, into healthy daughters that can then reproduce in the same way. But while such asexual reproduction is simple and direct, it gives rise to offspring that are genetically identical to the parent organism. Sexual reproduction, on the other hand, involves the mixing of genomes from two individuals to produce offspring that are genetically distinct from one another and from both their parents. This mode of reproduction apparently has great advantages, as the vast majority of plants and animals have adopted it. Sexual reproduction involves both diploid and haploid cells. Organisms that reproduce sexually are generally diploid. Each cell contains two sets of chromosomes, a maternal chromosome set and a paternal chromosome set, one inherited from each parent. The two parents, as members of the same species, have chromosome sets that are similar except for the sex chromosomes, specialized chromosomes present in some organisms that distinguish males from females. Each diploid cell, therefore, carries two copies of each gene, with the exception of genes carried on the sex chromosomes, which may be present in only one copy. What makes individuals within a species different from each other is that genes occur in variant versions, or alleles. Many different alleles of each gene are generally present in the collective gene pool of a species. This means that the two copies of any given gene in a particular individual are often somewhat different from each other and from those carried by other individuals. Thanks to sexual reproduction, Each new individual represents a new combination of alleles. Unlike the other cells in a diploid organism, the specialized cells that perform the central process in sexual reproduction, the germ cells, or gametes, are haploid. That is, they each contain only one set of chromosomes. Typically, two types of gametes are produced. In animals, one is large and non-motile and is referred to as the egg. The other is small and motile and is referred to as the sperm. Figure 1930. These haploid germ cells are generated when a diploid cell undergoes meiosis. During meiosis, the chromosomes of the double chromosome set are partitioned out, in fresh combinations, into single chromosome sets. The two different haploid gametes then fuse to make a diploid cell, the fertilized egg, or zygote, with a new combination of chromosomes. Figure 19.4. The zygote thus produced develops into a new individual with a diploid set of chromosomes that is distinct from that of either parent. For almost all multicellular animals, including vertebrates, practically the whole life cycle is spent in the diploid state. The haploid cells exist only briefly, do not divide at all, and are highly specialized for their function as gametes. They have to be freshly generated from diploid precursor cells within the organism. This precursor cell lineage, dedicated to the production of germ cells, is called the germline. The cells forming the rest of the animal's body, the somatic cells, ultimately leave no progeny. Figure 19.5. They exist, in effect, only to help the cells of the germline to survive and propagate. The sexual reproductive cycle thus involves an alternation of haploid cells, each carrying a single set of chromosomes, with generations of diploid cells, each carrying two sets of chromosomes. The mixing of genomes that characterizes sexual reproduction is achieved by fusion of two haploid cells to form a diploid cell. In this way, through cycles of diploidy, meiosis, haploidy, and cell fusion, old combinations of genes are broken up and new combinations are created. Sexual reproduction gives organisms a competitive advantage. The new combinations of alleles produced in each round of sexual reproduction are generated by a random process, and they are at least as likely to represent a change for the worse as a change for the better. Why, then, should an ability to try out new genetic combinations give organisms that reproduce sexually an evolutionary advantage over those that breed through an asexual process? This problem continues to perplex evolutionary geneticists, but one advantage seems to be that reshuffling the genes through sexual reproduction can help a species survive in an unpredictably variable environment. If two parents produce many offspring with a wide variety of gene combinations, the chance that at least one of their progeny will have the combination of features necessary for survival is increased. This may explain why even unicellular organisms, such as yeasts, intermittently indulge in a simple form of sexual reproduction. Typically, they switch on this behavior as an alternative to ordinary cell division when times are hard and starvation looms. Yeasts with a genetic defect that makes them unable to reproduce sexually show a reduced ability to evolve and adapt when they are placed in harsh conditions. Sexual reproduction may also be advantageous for another reason. In any population, new mutations continually occur, giving rise to new alleles, and many of these new mutations will be harmful. Sexual reproduction can speed the elimination of these deleterious alleles and help to prevent them from accumulating in the population. By mating with only the fittest males, females select for good combinations of alleles and allow bad combinations to be lost from the population more efficiently than they would otherwise be. According to this theory, which has been supported by some careful calculations of cost and benefit, sexual reproduction is favored because males can serve as a genetic filtering device. The males who succeed in mating allow the best, and only the best, collections of genes to be passed on, while the males who fail to mate serve as a genetic trash can, a way of discarding bad collections of alleles from the population. Of course, evolution has taken many twists and turns since sexual reproduction first arose, and, In social organisms especially, it has to be conceded that males may sometimes make themselves useful in other ways. Whatever its advantages, sexual reproduction has clearly been favored by evolution. In the following section, we review its central features, beginning with meiosis, the process by which the sex cells are formed. Meiosis and fertilization. Our modern understanding of the fundamental cycle of events involved in cell reproduction grew out of discoveries reported in 1888, when Theodore Boveri observed that the fertilized egg of a parasitic roundworm contains 4 chromosomes, whereas the worm's gametes, sperm and egg, contain only 2. This observation revealed that gametes are haploid. They carry only a single set of chromosomes. All of the other cells of the body, including the cells that give rise to the gametes, are diploid. They carry 2 sets of chromosomes, one derived from the mother and the other from the father. Therefore, sperm and eggs must be formed by a special kind of cell division in which the number of chromosomes is precisely halved. The term meiosis was coined to describe this form of cell division. It comes from a Greek word meaning diminution, or lessening. From Boveri's experiments on worms and other species, it became clear that the behavior of the chromosomes, which at that time were simply stainable microscopic bodies of unknown function, matched the pattern of inheritance, in which the 2 parents make equal contributions to determining the character of the progeny despite the enormous differences in size between egg and sperm. See Figure 19.3. This was the clue that led to the first realization that the chromosomes contain the material of heredity. The study of sexual reproduction and meiosis, therefore, has a central place in the history of cell biology. In this section, we describe the underlying cell biology from a modern point of view, focusing on the elaborate dance of chromosomes that occurs when a cell undertakes meiosis. We begin with an overview of how meiosis distributes chromosomes to the gametes. We then take a closer look at how chromosomes pair, recombine, and are segregated during the process, thereby shuffling the maternal and paternal genes into novel combinations. We also discuss what happens when meiosis goes awry. Finally, we consider briefly the process of fertilization, through which gametes come together to form a new and genetically distinct individual. Haploid germ cells are produced from diploid cells through meiosis. When diploid cells divide by mitosis in the ordinary way, they begin by duplicating their 2 sets of chromosomes precisely, so as to allow identical sets of chromosomes, a complete maternal set plus a complete paternal set, to be transmitted to each daughter cell, discussed in Chapter 18. Meiosis is different because only a single set of chromosomes, consisting of part of the maternal and part of the paternal chromosome sets, is finally parceled out to each gamete from the diploid starting cell. Yet, perverse as it might seem, given that the final outcome is a reduction in the number of chromosomes, meiosis begins with a single round of DNA replication that duplicates all of the chromosomes. The ultimate reduction in chromosome number occurs because this single round of replication is followed by 2 successive cell divisions. One might imagine that meiosis could occur by a simple modification of a normal mitotic cell division, in which DNA replication, S phase, was omitted. In principle, a single round of cell division would then produce 2 haploid cells directly. But, for reasons that are still unclear, this is not the way it is done. Meiosis in many species is a relatively long drawn-out procedure, taking very much longer than any mitosis. In a human male, for example, the process requires 24 days. In a human female, it can go on for decades. Meiosis begins in specialized diploid germline cells in the ovaries or testes. Each of these cells contains 2 copies of each chromosome, one inherited from the organism's father, the paternal homologue, and one from its mother, the maternal homologue. In the first step of meiosis, the chromosomes of the diploid cell are duplicated. As in any cell preparing to divide, these duplicated chromosomes remain attached to one another, like a set of conjoined twins. The next phase of the process is unique to meiosis. Each duplicated maternal chromosome finds and pairs with the duplicated maternal homologue. This specialized pairing ensures that the homologues will segregate properly during the subsequent cell divisions, so that each of the final gametes receives a complete haploid set of chromosomes. two successive cell divisions, called meiotic division 1 and meiotic division 2, now parcel out a complete set of chromosomes to each of the 4 haploid cells produced. Because the assignment of the homologs to each cell is random, the original maternal and paternal chromosomes are reshuffled into different combinations in the gametes that will eventually form from these haploid cells. During fertilization, 2 gametes will unite, generating a diploid zygote that is genetically distinct from each of its parents. See Figure 19.4b. The zygote then develops into a multicellular organism through repeated rounds of cell division and cell specialization. Thus, meiosis produces 4 cells that are genetically dissimilar and that contain exactly half as many chromosomes as the original parent cell. In contrast, mitosis produces 2 genetically identical daughter cells. We now discuss the molecular event of the meiotic cycle in more detail, beginning with a pairing of maternal and paternal chromosomes, a process that is central to the specialized form of cell division. Meiosis involves a special process of chromosome pairing. Before the cell divides, by either meiosis or mitosis, it first duplicates all of its chromosomes, as we have just noted. The twin copies of each fully replicated chromosome, called sister chromatids, at first remain tightly linked along their length. The way these replicated chromosomes are handled, however, differs in meiosis and mitosis. In mitosis, as we saw in Chapter 18, the replicated chromosomes line up in random order at the metaphase plate. As mitosis continues, the 2 previously joined sister chromatids then separate from each other to become individual chromosomes, and the 2 daughter cells produced by cytokinesis inherit 1 copy of each paternal chromosome and 1 copy of each maternal chromosome. Thus, both sets of genetic information are transmitted intact to both daughter cells, which are, therefore, diploid and genetically identical. The events that occur in the first meiotic cell division mirror the sequence of stages that a cell goes through in mitosis. In prophase, the replicated chromosomes condense. In metaphase, they align at the equator of the meiotic spindle, and in anaphase, they are segregated to the poles. For a review of these stages, see Panel 18.1, pages 626 to 627. The need to have the number of chromosomes during meiosis, however, makes an extra demand on the cell division machinery and leads to the first main difference between meiosis and mitosis. In Division 1 of meiosis, the replicated paternal and maternal chromosomes, including the 2 replicated sex chromosomes, pair alongside each other before they line up on the spindle. Figure 1916. This physical pairing of matched sets of chromosomes, called homologous chromosomes or homologs, is crucial because it enables the paternal and maternal chromosomes to be segregated to different daughter cells at this first division. For each chromosome, allocation of the maternal or paternal homolog to one daughter cell or the other is random. In this way, the original maternal and paternal chromosomes, with their different sets of alleles, are reshuffled into new combinations in each daughter cell of the first meiotic division. How the homologs and the 2 sex chromosomes recognize each other is still not fully understood. In many organisms, the initial association, the process of pairing, seems to be mediated by an interaction between matching maternal and paternal DNA sequences at numerous sites that are widely dispersed along the chromosomes. The structure formed when the duplicated chromosomes pair is called bivalent, and it contains 4 chromatids. Figure 19.7. The bivalent forms and is maintained during the long meiotic prophase, a stage that can last for years in some organisms. Crossing over can occur between maternal and paternal chromosomes. The picture of meiotic division 1 that we have just painted is severely simplified, in that it leaves out one crucial feature. In almost all sexually reproducing organisms, the pairing of the maternal and paternal chromosomes is accompanied by recombination, a process in which an exchange of DNA occurs between 2 identical or very similar nucleotide sequences. See Figure 631. In a diploid germline cell, recombination occurs during the long prophase of the first meiotic division. This recombination typically results in a physical swapping of homologous segments from the maternal and paternal chromosomes, an event known as crossing over. See Figure 630. Crossing over in meiosis is a complex process catalyzed by an intricate protein machine, and it depends on the formation of synaptonymal complex. As a duplicated homologous pair, this elaborate complex holds them together and aligns them so that genetic recombination can readily occur between the non-sister chromatids. The structure also serves to space out the crossover events along each chromosome. Each of the two chromatids of a duplicated chromosome can cross over with either of the two chromatids of the other chromosome in the bivalent. By the time that prophase ends, the synaptonymal complex has disassembled, allowing the homologs to separate along most of their length. But each pair of duplicated homologs is now held together by at least one chiasma, plural, chiasmata, the connection that corresponds to a crossover between two non-sister chromatids. Figure 19.8. The structure is named after the Greek letter chi, which is shaped like a cross. Many bivalents contain more than one chiasmata, indicating that multiple crossovers can occur between homologous chromosomes. See Figure 19.8b and C and Figure 19.19. On average, between 2 and 3 crossover events occur between each pair of human chromosomes during meiosis 1. Crossovers that occur during meiosis are a major source of genetic variation in sexually reproducing species. By scrambling the genetic constitution of each of the chromosomes in the gamete, crossing over helps to produce individuals with novel assortments of genes. Crossing over not only generates new combinations of maternal and paternal genes of individual chromosomes, but also plays a second important role in meiosis. By holding homologous chromosomes together during prophase 1, crossing over ensures that the maternal and paternal homologs will segregate from one another correctly at the first meiotic division, as we discuss next. Chromosome pairing and recombination ensure the proper segregation of homologs. In most organisms, recombination during meiosis is required for the correct segregation of the two duplicated homologs into separate daughter nuclei. The chiasmata created by crossover events have a crucial role in tying together the maternal and paternal homologs until the spindle separates them at anaphase 1. Before anaphase 1, the two poles of the spindle pull on the duplicated homologs in opposite directions, and the chiasmata resist this pulling. Figure 1910a. In so doing, the chiasmata help to position and stabilize bivalents on the metaphase plate. In addition to the chiasmata, which holds the maternal and paternal homologs together, cohesin proteins keep the arms of the sister chromatids glued together along their length. See Figure 19.8 and 18.15. The sister chromatids become unglued when the cohesin on the arms is degraded at the start of anaphase 1. This allows the newly recombined homologs to be pulled apart. Figure 1910b. The second meiotic division produces haploid daughter cells. The first meiotic cell division does not produce cells with a haploid amount of DNA. To achieve this goal, each cell now proceeds through a second round of division, meiosis 2, which occurs without further DNA replication and without any significant interphase period. A spindle forms, the chromosomes align at its equator, and the sister chromatids now separate to produce daughter cells with a haploid DNA content. In division 2 of meiosis, the kinetic cores on each pair of sister chromatids attach to the kinetic core microtubules pointing in opposite directions, as in an ordinary mitotic division. This configuration allows the individual chromatids to be drawn into different daughter cells at anaphase 2, Figure 1911. When the meiosis-specific cohesins that hold the sister chromatids together at the centromere are suddenly degraded, the chromatids separate. To summarize, meiosis consists of a single round of DNA replication followed by 2 cell divisions, so that 4 non-identical haploid cells are produced from each diploid cell that enters meiosis. Movie 19.1. In contrast, the ordinary mitotic cell cycle begins with a diploid cell and produces 2 identical diploid cells. The two processes are compared in Figure 1912. Haploid cells contain reassorted genetic information. Identical twins, which develop from a single zygote, are genetically identical. Otherwise, no two siblings are genetically the same. This is true because, even before fertilization takes place, meiosis has produced two kinds of randomizing genetic reassortments. First, as we have seen, the maternal and paternal chromosomes are shuffled and dealt out among the gametes during meiosis. Although the chromosomes are carefully distributed so that each gamete receives one and only one copy of each chromosome, the choice between the two copies is made at random. Thus, each gamete contains maternal versions of some chromosomes and paternal versions of others. Figure 1913a. The assortment is decided entirely by the way each bivalent is positioned when it lines up on the spindle during metaphase 1. Whether the maternal or paternal homolog is captured by the spindles from one pole or the other depends on which way the bivalent is facing when the microtubules connect with its kinetic core. See Figure 1910. Because the orientation of each bivalent at the moment it is captured is completely random, the assortment of maternal and paternal chromosomes is random as well. Thanks to this type of reassortment alone, an individual could in principle produce 2 to the n genetically different gametes, where n is the haploid number of chromosomes. Each human, for example, can in theory produce 2 to the 23, equals 8.4 times 10 to the 6 different gametes simply from the random assortment of paternal and maternal homologs that takes place in meiosis. The actual number of different gametes each person could produce, however, is much greater than 2 to the 23. This is because the crossing over that takes place during meiosis provides a second source of randomized genetic reassortment. Between 2 and 3 crossovers occur on average on each pair of human chromosomes per meiosis. This process puts maternal and paternal genes that are initially on separate chromosomes onto the same chromosome, as illustrated in Figure 1913b. Because crossing over occurs at more or less random sites along the length of a chromosome, each meiosis will produce 4 sets of entirely novel chromosomes. The reassortment of chromosomes in meiosis, together with the recombination of genes that arises from crossing over, provides a nearly limitless source of genetic variation in the gametes produced by a single individual. Considering that each human is formed from the fusion of 2 such gametes, one from the father and one from the mother, the richness of human variation that we see around us, even within a single family, is not at all surprising. Meiosis is not flawless. The sorting of chromosomes that takes place during meiosis is a remarkable feat of cellular bookkeeping. In humans, each meiosis requires that the starting cell keep track of 92 chromosomes, 23 pairs, each of which has duplicated, handing out one complete set to each gamete. Not surprisingly, mistakes do occur in the distribution of chromosomes during this elaborate process. Occasionally, homologs fail to separate properly, a phenomenon known as Nondisjunction. As a result, some of the haploid cells that are produced lack a particular chromosome, while others have more than one copy of it. Upon fertilization, such gametes form abnormal embryos, most of which die. Some survive, however. Down syndrome, for example, a human disease involving mental retardation and characteristic physical abnormalities, is caused by an extra copy of chromosome 21. This error results from non-disjunction of a chromosome 21 pair during meiosis, giving rise to a gamete that contains 2 copies of chromosome 21 instead of 1 copy. Figure 1914. When this abnormal gamete fuses with a normal gamete at fertilization, the resulting embryo contains 3 copies of chromosome 21 instead of 2. This chromosome imbalance produces an extra dose of the proteins encoded on chromosome 21 and thereby interferes with the proper development of the embryo. The frequency of missegregation in human gametes is remarkably high, particularly in the female. Non-disjunction occurs in about 10% of the meioses in human oocytes, giving rise to eggs that contain the wrong number of chromosomes, a condition called aneuploidy. Aneuploidy occurs less often in human sperm, perhaps because sperm development is subjected to more stringent quality control than egg development. If meiosis goes wrong in male cells, a cell cycle checkpoint mechanism is activated, arresting meiosis and leading to cell death by apoptosis, regardless of whether the segregation error occurs in the sperm or egg. Nondisjunction is thought to be one reason for the high rate of miscarriages, spontaneous abortions, in early pregnancy in humans. Fertilization reconstitutes a complete diploid genome. Having seen how chromosomes are parceled out during meiosis, we now discuss briefly how they come back together during the process of fertilization to form a new zygote with a complete set of chromosomes. Of the 300 million human sperm ejaculated during coitus, only about 200 reach the site of fertilization in the oviduct. There is evidence that chemical signals released by cells that surround the ovulated egg attract the sperm to the egg, but the nature of the chemoattractant molecules is unknown. Once it finds an egg, the sperm must migrate through a protective layer of cells and then bind to and tunnel through the egg coat, called the zona pellucida. Finally, the sperm must bind to and fuse with the underlying egg plasma membrane. Figure 1915. Although fertilization normally occurs by this process of sperm-egg fusion, it can also be achieved artificially by injecting the sperm directly into the egg cytoplasm. This is sometimes done in infertility clinics where there is a problem with natural sperm-egg fusion. Although many sperm may bind to an egg, only one normally fuses with the egg plasma membrane and introduces its DNA into the egg cytoplasm. The control of this step is especially important because it ensures that a fertilized egg will contain 2, and only 2, sets of chromosomes. Several mechanisms prevent multiple sperms from entering an egg. In one mechanism, the first successful sperm triggers the release of a wave of Ca2+ ions in the egg cytoplasm. The Ca2+ in turn then triggers the secretion of enzymes that cause a hardening of the zona pellucida. This prevents runner-up sperm from penetrating the zona pellucida, thereby ensuring that in the race to fertilize the egg there is a single winner. To watch a fertilization-induced calcium wave, see movies 16.4 and 19.2. Once fertilized, the egg is called a zygote. The process is not complete, however, until the 2 haploid nuclei, called pronuclei, come together and combine the chromosomes into a single diploid nucleus. Fertilization marks the beginning of one of the most remarkable phenomena in all of biology, the process of embryogenesis, in which the zygote divides to produce large numbers of diploid cells that develop into a new individual. Mendel and the laws of inheritance. In organisms that reproduce without sex, the genetic material of the parent is transmitted faithfully to its progeny. Thus, offspring are genetically identical to a single parent. Before Mendel started working with peas, some biologists suspected that inheritance might work that way in people. Figure 1916. But though children resemble their parents, they are not carbon copies of either the mother or the father. For organisms that reproduce sexually, offspring tend to exhibit a mixture of traits derived from both parents. This fact helped early geneticists, scientists who study heredity, in their efforts to unravel the laws of inheritance. To understand the underlying principles of heredity, one needs to be able to track particular characteristics as they pass, or fail to pass, from parent to child, and those characteristics have to show some variation. If every blue-eyed parent gave birth only to blue-eyed children, generation after generation, it would be impossible, on the basis of studying those families, to go beyond the statement that blue eye color is inherited. Thanks to the mechanisms of meiosis just described, sex breaks up existing collections of genetic information, shuffles alleles into new combinations, and produces individuals that have different characters as a result. It is therefore not surprising that the study of inheritance via sexual reproduction provided the first crucial insight into the mechanisms of heredity. Humans, like other species, exhibit simple traits that can be followed from one generation to the next, such as whether an individual's earlobes are attached or pendulous, or whether a person can detect certain odors or flavors. Figure 1917. But our family sizes are small, and human development is so slow that it takes 40 years or more just to get a couple of generations of progeny to analyze. Humans, therefore, don't make the best experimental material for geneticists. The laws of heredity were instead unraveled in species that are easy to breed and that produced large numbers of offspring. Gregor Mendel, the father of genetics, chose to study peas, but similar experiments can be performed in fruit flies, worms, dogs, cats, or any other plant or animal that possesses characteristics of interest. We know now that the same basic laws of inheritance apply to all sexually reproducing organisms, from microscopic yeasts to peas to people. In this section, we describe the logic of genetic inheritance in sexually reproducing organisms. We shall see how the behavior of chromosomes during meiosis, their segregation into gametes that then unite at random to form genetically unique offspring, explains the experimentally derived laws of inheritance. But to begin, we discuss how Mendel, breeding peas in his monastery garden, discovered these laws in the 19th century. Mendel chose to study traits that are inherited in a discrete fashion. When designing an experiment to address a scientific question, selecting the right organism can be critical. Mendel chose to carry out his studies in pea plants. Peas are easy to cultivate quickly and large numbers can be raised in a small place, such as an abbey garden. Furthermore, Mendel could control which plants mated with which. Each flower on a pea plant contains both male and female structures, and left to their own devices, the plants normally self-fertilize. But Mendel found he could cross-pollinate the peas by removing the immature male parts from one flower and then fertilizing that emasculated plant with sperm, pollen, from another plant. Thus, Mendel could be certain of the parentage of every pea plant he examined. Equally critical for Mendel's purposes, pea plants were available in many varieties. For example, one variety has purple flowers, another has white. One variety produces peas with smooth skins, another produces peas that are wrinkled. Mendel chose to follow sets of traits, such as flower color and seed shape, that were distinct, easily observable, and, more importantly, inherited in a discrete fashion. Figure 1918. In other words, the plants have either purple flowers or white flowers, nothing in between. Mendel could disprove the alternative theories of inheritance. The breeding experiments that Mendel performed were straightforward. He started with stocks of genetically pure, true breeding plants. When true breeding plants self-pollinate, all of their offspring are of the same variety. If he were following seed color, for example, he would use plants with yellow peas that always produced offspring with yellow peas, and plants with green peas that always produced offspring with green peas. Mendel took the unique approach of studying each trait one at a time. His predecessors had focused on whole organisms that varied in many traits, and they often wound up trying to characterize offspring whose appearance varied in such a complex way that the progeny could not easily be compared with appearance. In a typical experiment, Mendel would take two of his true breeding varieties and cross-pollinate them. Then he would record the inheritance of the chosen trait in the next generation. For example, Mendel crossed plants producing yellow peas with plants producing green peas. In this case, he discovered that the resulting hybrid offspring, called the first filial or F1 generation, all had yellow peas. Figure 1919. A similar finding held true for every trait he followed. The F1 hybrids all resembled only one of their two parents. From his experiment with green and yellow-seeded pea plants, Mendel could conclude that seed color is not passed down by blending. If it were, the peas in the F1 generation would have all been yellowish-green. On the surface, though, the results seem to support a theory of uniparental inheritance, which posits that the appearance of the offspring will match one parent or the other. Had Mendel stopped there, observing only the F1 generation, he might have developed some mistaken ideas about the nature of heredity. Fortunately, he took his breeding experiments to the next step, mating the F1 plants to one another, or allowing them to self-fertilize, and examining the results. Mendel's experiments were the first to reveal the discrete nature of heredity. Mendel's experiments with the hybrid F1 plants were designed to address an obvious question. What had happened to the traits such as green pea color, that disappeared in the F1 generation. See Figure 1919. Did the plants bearing green peas fail to make a genetic contribution to their offspring? To find out, Mendel allowed the F1 plants to self-fertilize. If the trait for green peas, for example, had been lost, then the F1 plants would produce only plants with yellow peas in the next F2 generation. He used a large sample size and kept an accurate count of the results, and he found that the disappearing trait returned. Although 3/4 of the offspring in the F2 generation had yellow peas, 1/4 had green peas. See Figure 1919. This result definitively laid to rest the theory of blended inheritance. There is simply no way that blending could explain how a cross between one yellow pea plant and another yellow pea plant could yield plants with green peas. But the data also gave Mendel a clue as to what was going on. Although the green pea plant disappeared temporarily in the F1 generation, it reappeared in F2. This means that at least some of the F1 plants must have still harbored a factor that specifies green peas. It was just hidden somehow. Mendel saw the same type of behavior for each of the other 6 traits he examined. To account for these observations, Mendel proposed that the inheritance of traits is governed by hereditary factors, which we now call genes, and that these factors act like discrete particles that remain separate instead of blending. Furthermore, he suggested that genes come in alternative versions that account for the variation seen in inherited characteristics. The gene dictating seed color, for example, exists in 2 flavors, one that directs the production of yellow peas and one that directs production of green peas. Such alternative versions of a gene are what we now call alleles, and the whole collection of alleles possessed by an individual, its genetic makeup, is called its genotype. Mendel reasoned that for each characteristic, a plant must inherit 2 copies, or alleles, of each gene, one from its mother, one from its father. The true breeding parental strains, he theorized, each possessed a pair of identical alleles. The yellow pea plants possess 2 alleles for yellow peas, the green pea plant 2 alleles for green peas. An individual that possesses 2 identical alleles is said to be homozygous for that trait. The F1 hybrid plants, on the other hand, had received 2 dissimilar alleles, one specifying yellow peas and the other green. These plants were heterozygous for the trait of interest. The appearance, or phenotype, of the plant depends on which versions of each allele it gets. To explain the disappearance of one of the traits in the F1 generation, Mendel supposed that for any pair of alleles, one allele is dominant and the other is recessive or hidden. The dominant allele, whenever it is present, would dictate the plant's phenotype. In the case of seed color, the allele that specifies yellow peas is dominant. The green pea allele is recessive. One important consequence of heterozygosity and of dominance and recessiveness is that not all of the alleles that an individual carries can be detected in its phenotype. Humans have about 25,000 genes, and each of us is heterozygous for a very large number of these. Thus, we all carry a great deal of genetic information that remains hidden in our personal phenotype, but that can turn up in future generations. Each gamete carries a single allele for each character. Mendel's theory that for every gene, an individual inherits 1 copy from its mother and 1 copy from its father raised some logistical issues. If an organism has 2 copies of every gene, how does it pass only 1 copy of each to its progeny? And how do these gene sets come together again in the resulting offspring? Mendel postulated that when sperm and eggs are formed, the 2 copies of each gene present in the parent separate so that each gamete receives only 1 allele for each trait. Thus, each egg, ovum, and each sperm, pollen, receives only 1 allele for seed color, either yellow or green, 1 allele for seed shape, smooth or wrinkled, 1 allele for flower color, purple or white, and so on. During fertilization, sperm with sperm with one or the other allele unites with an egg carrying one or the other allele, restoring the two copies of the gene for each trait in the fertilized egg or zygote. Which type of sperm unites with which type of egg at fertilization is entirely a matter of chance. This principle of heredity is laid out in Mendel's first law, the Law of Segregation, which states that the two alleles for each trait separate or segregate during gamete formation, and that they then unite at random, one from each parent, at fertilization. According to the Law of Segregation, the F1 hybrid plants with yellow peas will produce two classes of gametes. Half the gametes will get a yellow pea allele and half will get a green pea allele. When the hybrid plants self-pollinate, these two classes of gametes will unite at random. Thus, an ovum with a green pea allele has an equal chance of being fertilized by a pollen grain carrying a green pea allele or a yellow pea allele. The same is true for an ovum carrying a yellow pea allele. Thus, 4 different combinations of alleles can come together in the F2 offspring. Figure 1920. 1/4 of the F2 plants will receive 2 alleles specifying green peas. These plants will obviously produce green peas. 1/4 of the plants will receive 2 yellow pea alleles and will produce yellow peas. But 1/2 of the plants will inherit 1 allele for yellow peas and 1 allele for green. Because the yellow allele is dominant, these plants, like the heterozygous F1 parents, will all produce yellow peas. All told, 3/4 of the offspring will have yellow peas and 1/4 will have green peas. Thus, Mendel's law of segregation explains the 3-to-1 ratio that he observed in the F2 generation. Mendel's law of segregation applies to all sexually reproducing organisms. Mendel's law of segregation explained the data for all of the traits he examined in peas. He was also able to replicate his findings with corn and beans. But his rules governing inheritance are not limited to reproduction in plants. Mendel's concept of the gene and his law of segregation can be generalized to all sexually reproducing organisms, including humans. Let's consider a human phenotype that reflects the action of a single gene. The major form of albinism, type 2 albinism, is a rare condition that is inherited in a recessive manner in many animals, including humans. In other words, like the pea plants that produce green seeds, albinos are homozygous for the recessive allele A of a particular gene. Their genotype is a. The dominant allele, capital A, of the gene encodes for an enzyme involved in making melanin, the pigment that is responsible for most of the brown and black color present in hair, skin, and the retina of the eye. The recessive allele codes for a version of this enzyme that is less active or completely inactive. Without this enzyme, albinos have white hair, white skin, and pupils that look pink because the lack of coloration reveals the red color of the hemoglobin present in blood vessels in the retina. If a type 2 albino man, genotype a, has children with a type 2 albino woman, whose genotype is also a, all of their children will be albino, a. However, imagine that a non-albino man marries and has children with an albino woman. We will assume that the man is homozygous for the dominant capital A allele. Albinism is quite rare, and if no one in his family has ever had the condition, this genotype is probably valid. Their children should also be normally pigmented. That is, none would be albino. This result mirrors Mendel's process between 2 breeding pea plants. The father would contribute a dominant capital A allele to each gamete, and the mother would contribute a recessive a. The offspring, with genotype capital A would all have the dominant phenotype. Figure 1921. If one of these children someday met and started a family with an individual of a similar genotype, a man or a woman who also has an albino parent, we would expect that their children would follow the pattern seen in Mendel's F2 generation. For every 3 normally pigmented children, there would be, on average, 1 albino child. Or, from the point of view of an individual child, each would have a 25% chance of receiving 2 recessive alleles. Of course, humans generally don't have families large enough to guarantee accurate Mendelian ratios. Mendel arrived at his ratios by breeding and counting thousands of peas for most of his crosses. Geneticists interested in following the inheritance of specific traits in humans get around this problem by working with large numbers of families, or with several generations of a few large families. To keep track of this type of information and to help draw out the pattern of inheritance, geneticists prepare a pedigree that shows the phenotype of each family member for the relevant trait. Figure 1922 gives an example, illustrating an important practical consequence of Mendel's laws. It shows how first-cousin marriages create a greatly increased risk of producing children that are homozygous for a recessive deleterious mutation and so display a mutant phenotype. Alleles for different traits segregate independently. Mendel deliberately simplified the problem of heredity by performing monohybrid crosses, breeding experiments that focused on the inheritance of one trait at a time. He then continued his studies, next examining the simultaneous inheritance of two or more apparently unrelated traits. In the simplest situation, a dihybrid cross, Mendel followed the inheritance of two traits at once, seed color and seed shape, for example. For seed color, we have already seen that yellow is dominant over green. In the case of seed shape, round is dominant over wrinkled. What happens, Mendel wondered, when plants that differ in both of these characters are crossed? Again, he started with true breeding parental strains. The dominant strain produced yellow round peas. Its genotype is Y, R. The recessive strain produced green wrinkled peas. Lowercase y, lowercase r. One possibility is that the two characters, seed color and seed shape, could be transmitted from parents to offspring as a linked package. In other words, plants would always produce either yellow round peas or green wrinkled ones. The other possibility is that the seed color and seed shape would be inherited independently of one another, which means at some point plants that produce a novel mix of traits, yellow wrinkled peas or green round peas, would arise. Mendel bred the plants and kept a careful record of his results. As expected, the F1 generation showed a single phenotype. All plants produced peas that were yellow and round, but this would occur regardless of whether the parental alleles were linked. When these F1 plants were allowed to self-fertilize, the results clearly showed that each character is independently inherited. That is, the two alleles for seed color segregate independently of the two alleles for seed shape, producing four different pea phenotypes: yellow round, yellow wrinkled, green round, and green wrinkled. Figure 1923. Mendel tried his 7 pea characteristics in various pairwise combinations and always observed a characteristic 9:3 to 3:1 phenotypic ratio in the F2 generation. The independent segregation of each pair of alleles during gamete formation is known as Mendel's second law, the law of independent assortment. The behavior of chromosomes during meiosis underlies Mendel's laws of inheritance. So far, we have talked about alleles and genes as disembodied entities. As biologists, however, we are interested in heredity as more than a collection of mathematical ratios and probabilities. The likelihood that a pea plant will have purple flowers or that a child will be born an albino. We wish to understand how heredity works inside the sperm, the egg, and the resulting zygote. Mendel had assumed that genes are located in cells, but he didn't know that what they were made of or where they could be found. We now know that Mendel's factors, which we call genes, are carried on chromosomes that are parceled out during the formation of gametes and then brought together in novel combinations in the zygote at fertilization. Chromosomes therefore provide the physical basis for Mendel's laws. As we shall now see, their behavior during meiosis and fertilization explains Mendel's laws perfectly. During meiosis, as we discussed earlier, the maternal and paternal homologs and their genes that lie on them pair and then separate from one another on their way to being parceled out into gametes. These homologous chromosomes will possess different variants, or alleles, of many of the genes they carry. Take, for example, a pea plant that is heterozygous for the yellow pea gene, capital Y lowercase y. During meiosis, the chromosomes bearing the capital Y and lowercase y alleles will be separated, producing two types of haploid gametes, one that contains a capital Y allele and others that contain a lowercase y. Upon self-fertilization, these haploid gametes recombine at random to produce the diploid individuals of the next generation, which may be capital Y, capital Y lowercase y, or lowercase y. The meiotic mechanisms that drive the separation of alleles into gametes and the random recombination of gametes at fertilization give rise to exactly the result that Mendel's genetic laws describe. During meiosis, each set of paired homologs attaches to the spindle independently. This random arrangement of chromosomes on the metaphase spindle is reflected in Mendel's law of independent assortment, since genes on different chromosomes will be inherited independently. Although each gamete receives one, and only one, copy of each chromosome, it winds up with a random mixture of paternal and maternal homologs. See Figure 1913a. Figure 1924 diagrams the process for a pea plant that is heterozygous for both seed color, capital Y lowercase y, and seed shape, capital R lowercase r. The chromosome pair carrying the color alleles will attach to the meiotic spindle with a certain orientation. Whether the capital Y bearing homolog or the lowercase y bearing homolog is captured by the microtubules from one pole or the other depends on which way the bivalent happens to be facing at the moment of attachment. See Figure 1924. The same is true for the chromosome pair carrying the alleles for seed shape. Thus, whether the final gamete receives the capital Y lowercase y capital R, or lowercase y lowercase r combination of alleles depends entirely on which way the two chromosome pairs were facing when they were captured by the meiotic spindle, which has the same degree of randomness as a tossing of a coin. Chromosome crossovers can be used to determine the order of genes. Mendel studied 7 traits that were carried by 7 genes, each of which segregated independently of the others. It turns out that most of these genes are located on different chromosomes, which readily explains the random assortment he observed. But Mendel's observation that different traits assort independently does not necessarily require that the genes lie on different chromosomes. Genes that are far enough away from one another on the same chromosome will also sort independently because of the crossing over that occurs during meiosis. As we discussed earlier, when the duplicated chromosomes form bivalents and line up on the metaphase spindle, maternal and paternal homologs typically undergo several recombination events and thereby exchange genetic material. Such crossing over events can separate alleles that were formerly together on the same chromosome, causing them to segregate into different gametes. Figure 1925. We now know, for example, that Mendel's genes for pea shape and pod color are located on the same chromosome, but because they are far apart, they segregate independently. Of course, not all genes are inherited independently as per Mendel's second law. If genes lie closer together on a chromosome, they are likely to be inherited as a unit. For example, genes associated with red-green color blindness and hemophilia in humans are typically inherited together for this reason. By measuring how frequently genes are co-inherited, researchers can determine whether they reside on the same chromosome, and, if so, how far apart they lie. These measurements of genetic linkage have been used to map the relative positions of the genes on each chromosome of many organisms. Such genetic maps have been crucial for isolating and characterizing genes whose disruption by mutation is responsible for human genetic diseases such as cystic fibrosis. Mutations in genes can cause a loss of function or a gain of function. Mutations can be classified into a number of different basic categories. Figure 1926. Those changes that reduce or eliminate the activity of a gene are called loss-of-function mutations. An organism that receives 2 copies of a loss-of-function allele will generally show a mutant phenotype, one that differs from the normal, most commonly occurring, phenotype. The heterozygote, possessing 1 mutant allele and 1 wild-type allele, generally makes enough active gene product to function normally and retain the wild-type phenotype. Panel 19.1. page 674. Thus, loss-of-function mutations are usually recessive, because, for most genes, a halving of normal amount of gene product still leaves enough for the cell to function normally. As an example, in the case of Mendel's wrinkled peas, the gene that dictates seed shape codes for an enzyme that helps to convert sugars into branched starch molecules. The wild type, dominant allele, capital R, produces the active enzyme. The recessive, mutant allele, lowercase r, does not, because they lack this enzyme. Plants that are homozygous for the lowercase r allele contain more sugar and produce less starch than plants that possess the dominant capital R allele, which gives their peas a wrinkled appearance. The frozen peas available in the supermarket are wrinkled mutants, although the alleles they carry may not be the same one that Mendel used. Another class of mutant alleles produces proteins that are overreactive, or are active in inappropriate circumstances. Such gain-of-function mutations are usually dominant. For example, as we saw in Chapter 16, certain mutations in the gene encoding ROS, a protein involved in controlling cell growth and proliferation, generate a form of the protein that is always active and therefore overshadows the wild-type allele. The mutant ROS protein can stimulate cells to divide inappropriately, even in the absence of any growth signal. About 30% of all human cancers contain such dominant activating mutations in the ROS gene. Each of us carries many potential harmful recessive mutant alleles. As we saw in Chapter 9, mutations provide the fodder for evolution. Mutations can alter the fitness of an organism, making it either less or more likely to survive and leave progeny. Natural selection determines whether these mutations are preserved. Those changes that confer a selective advantage on an organism tend to be perpetuated, while those that compromise an organism's fitness tend to be lost. The great majority of chance mutations are either neutral, with no effect on phenotype, or deleterious. A deleterious mutation that is dominant, one that exerts its negative effects when present even in a single copy, will be eliminated almost as soon as it arises. If a mutant organism is unable to reproduce, the mutation that causes that failure will be lost from the population as soon as a mutant individual dies. For deleterious mutations that are recessive, things are a little more complicated. When such a mutation first arises, through some chemical accident in a DNA molecule, it will generally be present in only a single copy, and the carrier of the mutation will produce just as many progeny as other individuals. Many of these progeny will inherit a single copy of the mutation. They too will appear fit and healthy. But as they and their descendants begin to mate with one another, some individuals will inherit 2 copies of the mutant allele and show the harmful phenotype. If such a homozygous individual fails to reproduce, 2 copies of the mutant allele will be lost from the population. Eventually, an equilibrium is reached, where the rate of creation of new copies of the allele by newly occurring mutations balances the rate of loss through matings that yield homozygous individuals. As a consequence, many deleterious recessive mutations are present at a surprisingly high frequency in a population, even though individuals showing the deleterious phenotype are rare. Thus, children with the most common form of hereditary deafness, due to mutation in a gap junction protein, see Figure 2029, represent about 1 in 4,000 births, but about 1 in 30 of us are carriers of a loss-of-function mutant allele of the gene. Genetics as an experimental tool. The realization that chromosomes are the structures responsible for shutting our genes from one generation. The realization that chromosomes are the structures responsible for shuttling our genes from one generation to the next did more than demystify the basis of inheritance. It united the science of genetics with other disciplines: cell biology, biochemistry, physiology, and medicine. It also led to the discovery that genes are made of DNA. In this way, genetics has provided a path to new discoveries. By examining and manipulating DNA, we can begin to learn how our genes function together to create our phenotype, and how differences in genes underlie the differences between individuals. Increasingly, Knowledge of genetics enables us to diagnose and treat human diseases more accurately and helps us to decipher our similarities and differences, in relation both to other people and to other species. In this section, we outline the classical genetic approach to identifying genes and determining how they influence the phenotype of an organism. The process involves intentionally generating large numbers of mutant laboratory organisms and screening them to pick out those rare individuals that show a phenotype of interest. This genetic screen identifies bearers of mutation in the genes that govern that phenotype. By analyzing these mutant individuals and their progeny, we can track down and characterize the genes themselves, and, ultimately, work out the chain of cause and effect that leads from the genes to the phenotype. Modern technology has opened up additional ways to track down genes with significant functions. In the final part of this section, we discuss how we can examine DNA collected from human populations all over the world for clues to the genetics of complex traits and genetic diseases, those that are not simply governed by a single gene, as well as for hints about the evolution of our species. The classical approach begins with random mutagenesis. Before the advent of recombinant DNA technology discussed in Chapter 10, most genes were identified by observing the processes disrupted when the gene was mutated. The analysis begins with the isolation of mutants that have an interesting or unusual appearance. Fruit flies with white eyes or curly wings, for example. Working backward from this phenotype, one then determines the organism's genotype. This classic genetic approach, searching for mutant phenotypes and then isolating the genes responsible for them, is most easily performed in organisms that reproduce rapidly and are amenable to genetic manipulation, such as bacteria, yeast, worms, and fruit flies. Although spontaneous mutants can be found by examining extremely large populations, thousands or millions of individual organisms, the process of identifying interesting mutants can be made much more efficient by generating mutations artificially with agents that damage DNA, called mutagens. Different mutagens can generate different types of DNA alterations. Figure 1927. By treating organisms with mutagens, very large numbers of mutants can be created quickly and then screened for a particular defect of interest, as we will see shortly. Mutagenesis is a powerful tool for identifying genes and relating them to phenotypes in worms and flies. But how can we identify and study genes in humans? Unlike the organisms we have been discussing, humans do not reproduce rapidly, and intentional mutagenesis of humans is out of the question. Moreover, any human with a serious defect in an essential process, such as DNA replication, will die long before birth. There are two main answers to the questions of how we study human genes. First, because genes and gene function have been so highly conserved in the course of evolution, we can discover a great deal about human genes by investigating their counterparts in other organisms that are more suitable for experimentation. The corresponding human genes can be studied further in cultured human cells. Second, many mutations that are not necessarily lethal, such as the mutations causing deafness discussed above, have arisen spontaneously in the human population, and, indeed, have arisen many times over, because the human population is so large. Analyses of the phenotypes of the affected individuals, together with studies of their cultured cells, have provided many unique insights into human gene functions. Although such mutants are rare, they are very efficiently discovered because of a unique human property. The mutant individuals call attention to themselves by virtue of their peculiarities and by seeking special medical care. Genetic screens identify mutants deficient in specific cellular processes. A genetic screen typically involves examining many thousands of mutagenized individuals to find those few who show a specific altered phenotype of interest. For example, to search for genes involved in cell metabolism, one might screen mutagenized cells to pick out those that have lost their ability to grow in the absence of a particular amino acid or other nutrient. A problem arises, however, if we wish to study genes that are required for fundamental cell processes, RNA synthesis or cell cycle control, for instance. Defects in such genes are usually lethal, which means that special strategies are needed to propagate a stock of individuals that carry the interesting mutations. If you cannot breed the mutants, you cannot use them to study the gene.